So the first picture here shows you a serious red staining of failing on the right-hand side in comparison to non-failing myocardium. And you can appreciate here stained in yellow, and this is the same magnification on both sides, this enormous myocyte hypertrophy, and stained in red, you can appreciate the deposition of interstellar extracellular matrix in the interstitium, which we term fibrosis. So today I would like to share with you some recent work of my group on the role of microRNAs in both processes. In particular, I would like to transport the message that fibrosis, as we appreciate it typically as a process secondary to myocyte damage, can also be a primary driver of myocyte hypertrophy and remodeling itself. So the second introductory slide shows you here the classic cardiomyocyte hypertrophy assay, which many of us perform typically in vitro. So these are neonatal red cardiomyocytes stained here with a myocyte-specific marker alpha-actinin. Nuclei are here stained with DAPI in blue. And down here you see an automated edge detection algorithm to quantify the size of these cells. And here in the middle column you see what happens if we take a cell culture medium which has simply been conditioned by cardiac fibroblasts for 24 hours, and you see a rather clear-cut increase of the myocyte size, and this here is the quantification, and the positive control is 50 micromolar of the alpha-1 agonist phenylephrine, which is a known strongest known inducer of myocyte hypertrophy. So these are data from our lab, but in principle this has long been known that fibroblast conditioned medium by itself can exert a strong pro-hypertrophic effect, but this is often ignored in the field. The second thing which you can appreciate here, despite our efforts here to obtain really the purest possible neonatal cardiomyocyte cultures, I think we are already quite good, but you still see these red cells, and these are automatically detected as DAPI and nuclei that are not surrounded by alpha-actinin. So we have to keep in mind that all our hypertrophy assays in vitro, which are performed on neonatal myocytes, typically contain 5–10%, in some labs 30% of fibroblasts. So often we treat actually mixtures. So this is a bit too busy slide about the biology of microRNAs, and I think I can in principle shorten it. I expected it in the first talk, but I briefly go over it. So here on the left-hand side, depicted in blue, you can see a very simplified scheme of gene expression for protein coding genes. So we have here the promoter, we have the transcription here, and then we have the translation at the ribosome here in light blue of the messenger RNA, and here we have the protein. So what you have to know, and you are all aware of it, that probably the majority of all proteins are under significant control by endogenous non-protein coding RNAs. And a subspecies of these, and that's probably only the tip of the iceberg, of these non-coding RNAs are the microRNAs. These are of course genetically encoded. They are here transcribed, processed. We don't need the details here. They're processed by dicer then in the cytoplasm. We have two strands here depicted in red, the mature strand, and the other strand is typically rapidly degraded, which we term the star strand. In recent times it becomes obvious that the star strand, which is partially an antisense sequence, but not completely, to the mature strand, also has its own biological functions, and interestingly, in some instances, even an opposite function than the mature strand. But here we will focus, as most of the field so far, on the mature strand. This is incorporated into the RISC complex. The argonaude proteins are the most prominent constituent of that complex, and that then finds the 3' untranslated region of a messenger RNA, and the microRNA can then lead to repression of translation or to mRNA degradation. So the net effect is the same, that the microRNA inhibits the protein levels of its target. Typically, these effects are very subtle in the 20–30% range, and they are still quite powerful, as you know, and the reason of that is probably that they're targeting whole protein networks. So the first piece of data is very shortly what I would like to discuss with you, some work from Claudia Jenscher, PhD student in the lab, who set up similar but at a lower scale, as we heard by Mark McCullough, a screening approach in primary cardiomyocytes. So what she does is that she has highly purified, but not 100%, as you saw, neonatal red cardiomyocytes, which are cultivated on 96 well plates and are transfected with a microRNA library, very similar as we heard by Anna Olalio. And then segmentation analysis is carried out as you have seen it just. And the primary readout here is myocyte hypertrophy. So this is how our cultures here look like. Here on the left-hand side, alpha-actinine here as a myocyte-specific stain, P4HB here as a fibroblast marker, and nuclei stapi. And here you can see the automated image segmentation, very nice identification of individual cell area. And that's our primary readout here. And we have a very tight control of non-myocyte contamination. So here's a close-up. You've seen that already. Here's a classic hypertrophic response here. And here's a quantification of that on a few thousand cells. And it's a very robust assay with little variation. So the next slide here shows the principal result here of Claudia's work. So she identified quite a few microRNAs which are capable of inducing a rather strong, here's a twofold strong pro-hypertrophic effect. Here is the analysis if she restricts it to the 50 top expressed microRNAs, here the pro-hypertrophic ones, here anti-hypertrophic microRNAs. And this is highly reproducible. Here you see on the left-hand side an alternative repetition of these of this screen which had been performed in triplicate. And here you see in each case a radioactive assaying for protein synthesis, and actually the results here are highly reproducible for these pro-hypertrophic microRNAs. And if we use antimeres, we get actually rather modest effects, but many of them are also reach statistical significance then in the opposite direction. Interestingly, I'm not going into details here because this is very recently published, but we find very little correlation between differential expression of these pro-hypertrophic microRNAs in a TAC model, for example, of cardiac hypertrophy, and their pro-hypertrophic effect. So in other words, I think it makes sense to include these unbiased functional screens and not only base a candidate microRNA approach on the basis of differential expression in disease, which is, of course, an easily accessible and classic approach. So I would like to switch gears and now come to our primary work on fibroblasts. So what we've already just heard in the last talks is that microRNAs can also have cell type specific expression patterns. So here Simon Lyers-Eder in the lab took the challenge, or did the experiment, that he isolated adult fresh primary cells, fibroblasts and myocytes from healthy mouse hearts and from mice expressed to TAC. And then he carried out a microRNA array analysis and tried to identify microRNAs which are specific for myocytes, failing non-filing, and for fibroblasts. So here on the left-hand side, he used a rather weak criteria here, so twofold enrichment versus whole myocardium. And this is here the picture from the myocyte, and some of these microRNAs are extremely specific. Yeah? So microRNA 133 are microRNAs which are similar, like the myosins, extremely specific for myocytes. The picture is very different here for fibroblasts. We find clearly enriched fibroblast microRNAs, but not to an extent of enrichment which we find in myocytes. This is number one. The second result is here, I find quite remarkable that this pattern here is so different. Which I think is again a hint, what we also know from the literature, that the fibroblasts of the fibrotic heart are probably of very different origins, a very inhomogeneous population. So, which are now the top myocyte-specific microRNAs? These are the usual subsets. This essentially confirms what has been shown in the literature. This is not so clear for the cardiac fibroblasts, and 223, 125A, B, MIR21, and LET7B are here the top candidates for the fibroblasts. So many of fibroblast-enriched microRNAs are also functionally effective. These are very preliminary data here, which assay for fibroblast proliferation and for collagen secretion, and you can appreciate that several of these microRNAs seem to be potent in regulating fibroblast biology. So I come to briefly now go over two slides of published data on MIR21 from our group, and then come to more recent work on MIR21 and its putative mechanism of action. So the bottom line of our previous work on MIR21 is that we found it, and we saw it again with Mauro Giacca, that we found it to be very strongly upregulated, probably the most strongly upregulated microRNA in the diseased mammalian myocardium, and we find it to de-repress ERK-MAP kinase activity. And that de-repression occurs through inhibition of the endogenous MAP kinase inhibitor SPROUT1 and probably many other target proteins as well. The principal effect which we found on the fibroblast is an enhanced fibroblast survival, and we found it to strongly regulate cardiac fibrosis. So these are data from originally from Carina Gross. At that time, there were no commercial arrays available, but Mirko Castoldi and Martina Muckenthal at the EMBL in Heidelberg had set up the first custom-made microRNA arrays, and they were kind enough to hybridize for us RNA from a mouse model of cardiac failure from early, moderate, and late disease stage, and you see a progressive deregulation of the microRNA expression pattern. And marked in red here is MIR21, which we found consistently to be upregulated in mouse, but also in human cardiac failure. So one of probably the key experiment here from our early work here on MIR21 is depicted here. We used an anti-MIR, as we heard already, an 22 nucleotide long anti-MIR to inhibit endogenous MIR21. And we use a tag model here on the left-hand side. Here you see the original northern blot. And so what Carina found here is that if we inject this anti-MIR 3 consecutive days after the tag operation, and then take out the myocardium 18 days later, so 3 weeks in total, we find complete absence of that signal. And that is from non-consecutive lanes of a single northern blot. We also have much higher N numbers actually for every animal which we had treated here by quantitative real-time PCR, and the effect is really dramatic, and in the meantime has been reproduced by many groups. So these anti-MIR effects can be very long-lasting in vivo, surprisingly long-lasting. The principal effect here you can see of that treatment, you can see here on the lower side. You see in the upper row strong increase of interstitial fibrosis here on the tag model, and a complete inhibition here, prevention of fibrosis formation with the anti-MIR. And interestingly, also myocyte hypertrophy was clearly inhibited here by inhibition of MIR21. This is a simple prevention assay, which is often easier, of course, to achieve effects. What's more challenging is a therapeutic setting. So what we've done here, we did the tag operation, waited for 3 weeks. So if you do it strong enough, you already have some degree of fibrosis, cardiac dysfunction, as you saw. And then we did the treatment or the control. And then we looked at fractional shortening function here on the left-hand side, and you can appreciate the progressive course of the disease has been at least ameliorated. And left ventricular diameter, so the progressive dilatation, could also be partly rescued. So to sum up that early work, we suggest that miR-21 is upregulated in cardiac fibroblasts in the failing heart. That leads to an increase of ERK-MAP kinase activity, fibroblast survival, and that is a critical driver of fibrosis and overall cardiac remodeling and dysfunction. And with anti-miR-21, we can interfere here. So this work has now been taken by other groups to numerous settings of fibrosis. I just would like to summarize here. The first paper here was that inhibition of miR-21 prevents pulmonary fibrosis. So bleomycin, an anti-cancer agent, which is the very severe side effect of pulmonary fibrosis. This can be prevented in the mouse model. Then inhibition of miR-21 prevents kidney fibrosis. That has been shown last year. And very recently, it has been shown that knockout of miR-21 in science translational medicine could prevent kidney fibrosis. So it appears that this is not a mechanism specific for cardiac disease, but perhaps for many instances of organ fibrosis, which are clinically not acceptable at the point. So there was actually also one study which claimed that inhibition of miR-21 in cardiac fibrosis was completely ineffective. And this, of course, caused a severe headache and concern with Thomas Thume and myself, which hold major responsibility here for this original study. And so what in that paper was done is that an 8-mer, so an 8-nucleotide microRNA antagonist was used to inhibit miR-21. So what we did is a head-to-head comparison of exactly the same chemistry of the 8-mer. This needs to be LNA, otherwise it wouldn't hybridize at all, but that's not the critical point. The critical point is that it's only 8 nucleotides long. And we compared it to our original cholesterol-modified anti-mer, and then we used a cholesterol-free chemistry with these modifications here. So the bottom line is that we could completely confirm the inefficacy of the 8-mer. So the 8-mer you can inhibit for 48 hours in vivo. It's taken up, no problem. It cleans the northern blot signal for 48 hours, but then we find it's probably rapidly peed out, but we find that the miR-21 repressive effect, it's completely gone after a few days. And if you do the experiment for 3 weeks, with the 8-mer, we find no therapeutic effect whatsoever. It's very similar to PBS, but the cholesterol and the cholesterol-free chemistry clearly had therapeutic effects on hypertrophy, fibrosis, and cardiac function. So I would like to share now with you additional data which are actually not from our group, but which just were published a few weeks ago in the Journal of Cell Biology, which I find are therapeutically most interesting. And what happens here is that these people look at skeletal muscle fibrosis, Aditya et al. And what you observe, if you lacerate, if you cut a muscle and if you hear the repair response, you typically observe fibrosis similar as we see it in chronic myocardial failure. And they found strong upregulation of miR-21. And then they saw that anti-miR inhibited that fibrosis, and actually overexpression of miR-21 could promote fibrosis. So far, interesting, but not entirely exciting now, given what I've told you again. But now I think comes the interesting part. What they've now done is they switched to a model of chronic established fibrosis. And they looked at a mouse model of Duchenne's muscular dystrophy. And you know that at least in the diaphragm, you get a pathology which is very similar to the human disease. And then they look here at 12 months of age of these animals. So very chronic established tissue fibrosis. And as you know, in medicine, this is regarded as completely irreversible, and you can't do anything about it, and the patient has a very poor prognosis. So what they have done, they took these aged MDX mice, and then they used a couple of weeks of treatment with anti-miR-21 and saw a very strong regression here of interstitial fibrosis in these aged mice. And this is not only here histology, but you can also appreciate that they did here quantitative collagen and fibronectin expression, and so very significant effects here. So The conclusion is that miR-21 control of tissue fibrosis is, I think, a very promising therapeutic principle and might also hold true for other organs. So in the meantime, we have tried to work more on the downstream mechanism of miR-21. I'd like to share with you unpublished data on SPROUT1. So SPROUT1 has been described in other organs as a very potent and specific inhibitor of ERK-MAP kinase before, but not in the heart. I should like to say before I finish with my last 3 data slides that we certainly know that miR-21 has a variety of other targets which we and others have identified. So what about SPROUT? If miR-21 significantly acts through SPROUT to control cardiac fibrosis and hypertrophy, you would need to postulate that knockout of SPROUT has a similar effect than overexpression or high expression of miR-21. So these are now the first set of data. So these are homozygous knockouts for SPROUT, which were generated by Gail Martin at UCSF and which kindly provided these animals to us. And we find that we saw myocyte hypertrophy and some degree of interstitial fibrosis. We are the colony is not so aged at that point, but here's the male and female mice with, I think, reliable N numbers now, and there's a clear-cut increase of cardiac hypertrophy. So far, so good. We can use also a Laxet knock-in generated by Gail Martin's lab to more specifically locate SPR1 expression. And so we stain here for beta-gal for Laxet and to identify the cell type which expresses SPROUT1, and you can clearly see that here's co-staining with troponin for myocyte that SPROUT is expressed exclusively in non-myocyte cells. And as we heard already, also from Dr. Campogrossi, it's not very easy to specifically stain for fibroblasts, especially not in tissue. I think it was more convincing what we saw from Piero Anversa. But we, at least with Vimentin, we seem to have a clear-cut overlap with fibroblasts. So how does that look in isolated cells? That's, of course, more easy. If we isolate adult myocytes and adult fibroblasts, an assay here for SPROUT, there's a very strong enrichment here in the fibroblasts, and there are 3 isoforms expressed in the heart, but 2 and 4 are quantitatively negligible. So Markus Buss now started a work first in Gail Martin's lab, and that's now continued by Fabrice Schaffer in the lab, where he tries to knock out SPROUT1 from using a floxed allele, specifically non-myocytes. And as you know, the recent discussion with the periostin and FSP1 is a very complicated Cree lines. And this is probably an even more complicated one, but Markus Bussen digged that out. That's an early paper here where they used the Wnt antagonist Frisbee promoter to drive GRE, and they found a clear-cut non-myocyte expression here. And these are here the first data of and that's my last data slide. These are here the first data. We clearly see some degree of fibrosis here, perivascular at the point, but at least a tendency towards myocyte hypertrophy. Currently, we are using the WT1 Cree line to repeat this experiment. So I should like to thank the people from my group participating here, and also our host here, Mauro Giacca and Lorena Centellin, for providing AVs for several microRNAs. And thank you very much for your attention.
Thank you very much for